1% of life born, they have chromosomal abnormality. Chromosomal abnormalities or aberrations موجودين up to 27% in individuals, for example, with sex reversal, as we will see, or puberty anomalies. Up to 2/3 of spontaneous miscarriages, if you look at the fetus or the embryo, you will see that the reason was chromosomal abnormality. 5% of couples who have a history of multiple pregnancy loss or multiple miscarriages is due to chromosomal anomalies. The majority of cancers, even liquid cancers or solid tumors, are driven by chromosomal anomalies. Up to 2/3 of embryonic death, which happens before 5 weeks of age, are due to chromosomal anomalies, and those are considered unrecognized pregnancy. And the mother, she doesn't know necessarily that she's pregnant because the loss of pregnancy happens at an earlier stage. Infant and childhood death, 5 to 7%. They're born, but with birth defects, up to 8%. Congenital heart defects, 13%, are because of chromosomal anomalies. All right. So cytogenetics is the field of studying the chromosomes and chromosomal abnormalities. So you can perform cytogenetic studies to understand where the location of the abnormalities happening on the chromosome and what genes are involved. You can also recognize the chromosomal changes and the response to treatment. The next question is, which tissues, ايش هم الtissues اللي used to do chromosomal studies? Well, it depends on the disease. The easier tissue to do chromosomal studies through peripheral blood. So the lymphocytes, you can study the chromosomes by collecting blood from the patient, looking at the lymphocytes. Okay, so let me ask you a question here. Why lymphocytes? Why, for example, not erythrocytes? ممكن حدا يحاول يجاوب؟ ليش لما نعمل chromosomal study بنطلع على الكروموسومز بالليمفوسايتس مش بالريد بلاد سيلز؟
دكتور لأنه الوايت blood cells هي responsible for attacking disease tissues or foreign pathogens, which in this case could be chromosomal dysfunctions.
Not really, but let's discuss it. The structure of the blood cells اللي عندهم chromosomes هم الوايت blood cells ولا red blood cells؟ أريد إيش رأيك؟
Red blood cells doesn't have a nucleus.
بالضبط. Red blood cells, remember, they don't have nuclei. During their differentiation, they lose the nuclei. لأنه all what you care about as a red blood cell, you care about loading as much oxygen through hemoglobin. And the chromosomes are in the nuclei. Therefore, you would need to study white blood cells with lymphocytes if you want to do chromosomal studies. Even though the blood is the least invasive with chromosomal testing, but sometimes you would need to do chromosomal testing by more invasive procedures to access certain tissues. For example, certain cancer blood malignancies or blood cancers, you would need to study what's happening with the chromosomes in the bone marrow. For example, if there is pregnancy, بالultrasound or markers, you would see an abnormal finding, and you want to further pursue and understand, you might want to do chromosomal testing للفيتس أو الإمبريو من خلال collecting chorionic villi, هذا invasive procedure, أو collecting amniotic fluid اللي هو fluid bathing the baby هذول tissues من actual extra embryonic tissues that have the same genetic complement بالفيتس أو الembryo. Sometime you will need to have an organ biopsy. Okay, so this leads us to looking على الkaryogram, which is also known as karyotype. الkaryotype هو عبارة عن arrangement of the chromosomes from the bigger to the smaller على شكل pairs of chromosomes. احنا الhuman we have 46 chromosomes أو 23 pairs of chromosomes, one from each parent. Or you can arrange them على الkaryotype من أكبر واحد لأصغر واحد. The first 22 pairs are known autosomal chromosomes. The last pair, this pair here, is known as 6 chromosome. In males, it is XY, and in females, it is XX. Notice the chromosome, this is chromosome 1, and this is chromosome 1. So those are homologous chromosomes, one from each parent. واحد بيجي من father, paternal, and the other is maternal. Now, I want to bring your attention that the chromosome itself, this chromosome, consists of, even though you don't see it, they look very close to each other, but it's actually two sister chromatids. It's two sister chromatids. Now, the question is, at which stage in the cell cycle do you do a karyotype analysis or karyotype study? احنا عندنا الانترفيز وعندنا الام phase. So do we look at the chromosomes in the interphase or in the M phase? Can someone answer?
During the M phase or the prophase?
During the M phase, correct. لأنه بالinterphase بيكونوا الchromosomes diffuse على شكل chromatin, and actually you don't see them as the chromosome shape. If you remember, بالprophase, the chromatin starts to condense in a shape of chromosome, and it will keep condensing in prophase, and it will be the most condensed بالmetaphase. So as a matter of fact, what you're looking at here, you're looking at chromosomes in the metaphase. Why in metaphase? لأنه in anaphase, they are condensed as well. But in anaphase, sister chromatids will separate. يعني this entity will become two sister chromatids. بالanaphase, they will separate, and you want to see them before they separate. Okay, this is an ideogram, which is a cartoon representation of the 24 different chromosomes. As I told you, we have 22 autosomes, and we have X and Y. So we have 24 different chromosomes. So I mentioned a moment ago that the chromosomes are arranged from the biggest to the smallest. Now, to distinguish the chromosomes, it is based on their shape, centromere location, and banding pattern. If you look على البلاك bands, هذول الخطوط السوداء والبيضاء مختلفين اختلاف الكروموسومات. So you can identify the chromosome based on that. Now, when you do karyotyping, what you do basically, what you do is you arrest الليمفوسايتس. بوقف الليمفوسايت بالسيل سايكل بمرحلة الميتافيز. بعدين you stain them. تصبغهم صبغة اسمها جمزة, called Gimza stain. And you burst open, you rupture the membranes of the cell membrane with nuclear envelope. So you will allow the chromosomes to spread a little bit. And this is how they would look like under the microscope. Now, based on expertise and knowledge, you will start arranging the chromosomes, identifying each of them, or arranging them one by one. In the past, بالماضي, we used to use a scissor, تسعينات قبل, كان في مقص to cut around each chromosome. You print out the picture you see under the microscope. Using a scissor, you can cut each chromosome. بغرة أو بلاصق, you can tape them. Later on, the 2000s until today, you can use a software. You can use a mouse and draw a line to surround each chromosome. And using a software, you can give it a number, and the software will arrange the chromosome. Sometimes the softwares, the programs, they can guess. ممكن يحذروا which chromosome is which. But until now, it is based on human judgment. ما بعرف what will happen with the AI. Things will change. But until 2026, until now, it's based على human expertise. And as a matter of fact, those are my own chromosomes. This is my name here. So what I did, I collected blood, I stained my chromosomes, so I arranged them based on how they should appear بالkaryotype. Now, banding, هاي الbands, you see there are different types of banding. There is the G banding, there is the R, there is the C, Q, T, and Silverstein. Today, we will only talk about the first two banding patterns, which is the G and the R banding. The C with the Q, with T with Silverstein. Those are very specialized, they are very rarely used in the world. So I want you to be aware of the main banding pattern. And this is how different banding patterns look like. Let's start with the G banding. Well, the reason I want to start with the G banding because G banding is the most common banding pattern بالcytogenetics, بالkaryotyping. وهي routinely used clinical cytogenetic labs. So if you do a chromosomal testing, or if you hear of someone doing chromosomal testing, you would guess that individual had G banding performed by default. So G banding from the name GTG من جمزة, the way I remember it, الستين هي جمزة. Well, idea is simple. The idea is جمزة stain will bind to regions in the DNA that are AT rich, and it will not bind to regions in the DNA that is GC rich. Then the distribution of AT with GC varies across the DNA. Remember, each chromosome is a double-stranded DNA molecule before the S phase. After the S phase, each chromosome is two sister chromatids. Each of them is a double-stranded DNA molecule. They are identical to each other. Anyways, الجي باندنج binds على زي ما مكتوب هون ال AT rich region. Now the GC rich region is more common in coding genes. So DNA or the chromosome contains regions that are gene poor, يعني sequence of DNA, but there is no protein coding genes. Or regions تانيين that are gene rich. Now with gene rich regions, they will be rich بالnucleotide G and C. And the reason is the promoter itself, which is the sequence of DNA in a gene where the RNA polymerase with the transcription factors bind to transcribe. So that promoter region in the gene contains more GCs. GC. Well, GIMSA binds to AT. The AT is regions that are typically gene poor. Well, GC هو gene rich. لأنه ال AT is gene poor, then the DNA there typically is more condensed than a DNA regions that are gene rich. Well, condensed DNA is known as heterochromatin. So based on this discussion, GIMSA binds to GIMSA binds to AT, which is the heterochromatic gene poor regions, and it doesn't bind to E chromatin اللي هو less condensed region بالدي ان اي اللي that happens to be GC rich وgene rich. The number of bands across 23 pairs of human chromosomes حوالين 400 band, and each band itself is several megabases. Okay, يعني عدة ملايين من البيزز. This means, for example, if you have the disease اسمه دي جورج syndrome, دي جورج هو one of the common chromosomal deletions للchildren, affected children بهذا المرض, وهو deletion بهاي المنطقة بكروموسوم 22 بالكيو ارم اللي هي اللونج ارم اللي بتكون تحت. This region is deleted. الديليشن هذا إذا كان several megabases عدة ملايين من nucleotides, you can see it بالkaryotype. You can see it here if you are experienced. But if that deletion is really, really small, you will not see it بالkaryotype لأنه you will need an abnormality that is at least several million bases or more to be the disease causing to be captured بالkaryotype. So, and the dark bands you see here are حكيت لكم هم by Gimza that stains the AT-rich region that are gene poor or heterochromatin, with light bands that are the gene rich. So for example, if you look at chromosomes 19, you would see that chromosome 19 is light color, not dark color. والسبب that chromosome 19 is rich with genes. There are so many genes packed in this small chromosome. Okay. The other banding pattern I would like you to be aware of هي R banding. R is the reverse banding. من اسمها reverse, so therefore هي عكس ال G banding. إذا كان ال G banding اللايت bands هم للجين poor, sorry, اللايت bands هم للجين rich, والdark bands هم للجين poor, the R banding is the opposite, where the dark bands are the gene rich, the light bands are the gene poor. It uses Gimza stain, the same stain, but the only difference during the preparation of the sample, you heat the sample. When you heat the sample, the hydrogen bonds will weaken, وهذا الweakening بالhydrogen bonds راح يؤدي will lead to that Gimza stain, and instead of binding to AT, it will bind to GC. حكيت لكم ال GC هي gene rich. This is rarely used. One of the utilities of R banding. So for example, if you see an abnormality with G banding, but it's too small, or you're not 100% certain, مش متأكد 100% إنه that band is deleted, maybe you can reverse band to see if you flip the color, the banding pattern, if it would be more visible, right? So that is the R banding. Okay, now this leads us to discuss how the chromosomes are stained. We will look at the chromosomes, let's say from blood. With blood, we look at lymphocytes. Now in the lymphocytes, those are differentiated cells. هذول خلايا متميزة لهم شكل محدد بتميزوا تحت الميكروسكوب, well, they have a certain function. So those differentiated cells, typically they don't divide. The cells that divide هم will de-differentiated. But once they're differentiated, مجرد ما صار لهم شكل ووظيفة معينة, they don't divide. They are arrested, the cell cycle, they are arrested in interphase, typically بالG1. خلص الخلية صار لها شكل وظيفي بتوقف على الG1. So the first thing you need to do to induce a cell to undergo cell cycle. لأنه in G1 or interphase, as we discussed, the chromosomes are not visible, they are decondensed in a form of chromatin. So to visualize, you need to induce a cell to undergo a cycle. We want to arrest the cell during زي ما اتفقنا بالmetaphase where you typically see the chromosome. So the first thing you do here is you collect the tissue, let's say blood. You put that blood in a flask or in a petri dish. But then what you do, you add into the blood a chemical compound known as 5-hemagglutinin or PHA. This is known as a mitogen. الميتوجين هذا ال 5-hemagglutinin, its function, it will induce a cell to undergo cell cycle. تطلع من السيل arrest وتكمل بالسيل cycle. G1 S بالاس phase بصير عندي DNA replication وeach chromosome بصير عنده 2 sister chromatids. G2 and M, and the cycle continues. After you treat with 5-hemagglutinin, you wait for some time. The cells are undergoing in the cell cycle, and then you will treat with a mitotic inhibitor, with a chemical compound that will arrest the cell, will prevent the cell, or will stop the cell at metaphase. So typically, it's colchicine or colcimide. So now you have your cells arrested at metaphase, which is the phase where the chromosomes are condensed and visible under the microscope after you stain them. Next, you will centrifuge the cells in the tube, the blood cells, due to centrifugation process, the cells will precipitate in the pellet, and you will discard a supernatant. And after that, you will add hypotonic solution. The hypotonic solution هو a solution with lower salt concentration. And water will goes from the low salt concentration into a high salt concentration. So the water will go into the cells. Water goes into the cells will be swollen and they will be fragile. راح يكونوا منتفخين وهش يعني على وشك انهم ينفجروا. And then what you will do after you can wash the cells, بتشيل hypotonic solution, you put isotonic, and then you will drop the cells into a slide using a dropper. When you drop the cells with a dropper on a slide, the cells are swollen, they're fragile, and when they hit the slide surface, they will burst open. Well, the chromosomes will scatter. راح ينتشر الكروموسومات. There will be some space between them. So you can afterwards dip the slide in a jar with gemzestein. And afterwards you can fix and start cutting physically or digitally the chromosomes or arranging them in a shape of a karyotype. Okay. Now, if the chromosomes can be distinguished or divided based على centromere location. If the centromere is in the center, where it is hard to distinguish the P arm from the Q arm, P star stands for petite, which is supposed to be the shorter arm, while the Q stands for Q arm, which is supposed to be the longer arm. If the centromere is in the center, where the P and the Q, they look very similar, this is known as a metacentric chromosome, metacentric. If the centromere is shifted from the center, so the P arm looks significantly smaller than the Q arm, this is known as submetacentric. Example on metacentric chromosome number 2, example of submetacentric chromosome 18. If the P arm is shifted all the way to the end, then this is known as acrocentric. The acrocentric chromosomes, an example of that is chromosome 21. chromosome 21 the P arm of acrocentric chromosomes, all of them, كلهم عندهم the same structure, which is stalk and satellite. And this is how they look like under a light microscopy على 1000x magnification stained by Gimse. It is a centromere. Those are distant or telomeres. This is a metacentric chromosome. This one is submetacentric. And this one here is acrocentric. Based on this discussion, in this ideogram here, which chromosomes would you classify them as acrocentric? شو الكروموسومات اللي بتعتبروهم acrocentric بناء على الصورة اللي هون؟ تفضلي براءة.
بريء
آسف.
13 و14 و15 و21 و22.
Exactly. Chromosomes 13, 14, 15, 21, 22, those are the 5 acrocentric chromosomes. Y looks like acrocentric, but as a matter of fact, it's not. Only those 5 autosomes. زي ما حكيت لكم بريء 13, 14, 15, 21, and 22. And this is just another example showing الإيديوجرام والchromosomes based on centromere بنقسمهم meta, submeta, or acrocentric. The Q arm is longer than the Q arm. Now, each arm itself is divided into regions. For example, if I tell you this patient has a deletion in chromosome 7, Q arm, region 3, you tell me, okay, this is chromosome 7, this is the Q arm, and region 3 is here. So you're telling me that there is a deletion in this region, okay? And there are also subregions, as we will see in a minute. But let's talk a bit more about an acrocentric chromosome. The acrocentric chromosome, again, we have the stack and they have stack and satellite. And if you zoom in, this is how it would look like. Now, all of the chromosomes, including acrocentric, non-acrocentric, they have different genes on their arms, including the Q arms of acrocentric chromosome. لاحظوا هون ال AT rich الجين poor is stained dark والجين C rich الجين rich is stained light بالجي bending. And the telomeres اللي هم repetitive region راح نشوفهم بعدين at the end of the chromosome. Telomere. Now, T arm. Now let's talk about centromere. All of the chromosomes they have centromeres. والcentromeres عبارة عن satellite known as alpha satellite. الالفا satellite هاي بالcentromere هي عبارة عن repetitive sequence. لما نسمع كلمة satellite in DNA, think about repetitive sequence بتكرر. وtypically مش always but usually لما يخطر على بالي satellite or repetitive sequence I think of non-coding DNA. Question is, what is coding DNA? Coding DNA is a DNA that codes for a protein. Pseudotranscription, the RNA. The RNA is translated into protein, so it codes for a protein. Now with genes, we have احنا اللي human around 20,000 protein coding genes. We have also a little bit more than 20,000 non-coding genes. The non-coding genes, those genes, they're transcribed into RNA. بيعملوا RNA بس ال RNA هذا ما بصير protein. وطبعا most of the DNA معظم ال DNA doesn't have genes لا coding ولا non-coding. سموه gene desert. صحراء يعني ما فيها جينات. Now the centromere is non-coding. عبارة عن repetitive DNA. A sequence of DNA copy paste next to each other. We call it alpha satellite. There is also satellite بالبي arm of الacrocentric chromosome, just repetitive DNA that does encode لا protein coding genes ولا non protein coding genes. وعندنا non coding genes DNA هون بالبي arm that encodes بصير له transcription into rRNA. Remember, the only RNA that is translated into proteins is the messenger RNA. The ribosomal RNA or tRNA is not translated into protein, so the rRNA is non-coding gene. So ribosomal DNA هون and ribosomal DNA that is transcribed into ribosomal RNA. As a matter of fact, if you collect the RNA من blood or من أي tissue, the most common RNA in a tissue or in a cell is the rRNA. All of the 5 acrocentric chromosomes, 13, 14, 15, 21, and 22, they have the same P-arm structure, which means that all of the 5 P-arms of acrocentric chromosomes, they contain genes that encode for ribosomal RNA or satellite, non-repetitive sequence. Here's the satellite, and here is the stop where the rRNA exists. Now, it happens that if there is a deletion, if you look at a karyotype, and if an individual has a deletion in a P-arm of chromosome 13, this will not be clinically relevant, or it will not be disease-causing. Why? Can anyone guess? إيش إذا individual كان عنده البي arm deleted لكروموسوم 13 ما راح يؤدي لمرض.
Because it's non-coding.
It is coding فيها sorry, it is non-coding بس فيها genes اللي هم ribosomal DNA genes. لأنه
في sources ثاني بعوضه.
Yeah, exactly. لأنه even if the PR that encodes for ribosomal RNA genes is deleted, you have 4 other PRs that encode for the same genes. All right? So this is what is unique or special about the PR of acrocentric chromosomes. Back to banding, our discussion about the banding pattern. The banding pattern حكينا عن الكروموسوم جمزستين وعن بي ارم وكيو ارم والchromosomes are distinguished by the location of the centromere, the size of the chromosome, with banding pattern. What you're looking at here, you're looking at X chromosome. This is X, and this is X. هذا الكرتون وهذا actual karyotype for the same X chromosome. What's different is that this version of chromosome X is more condensed than this version. So as a matter of fact, you can extend a number of bands from 400 bands to double of that to 800 من خلال high resolution banding. High resolution banding, remember, the cell will start condensing the chromosome with a prophase. The chromosomes will be the most condensed al metaphase, and this is when the standard karyotyping is performed. You can do high resolution banding by arresting a cell before الميتافيز من خلال pro-metaphase. If you arrest a cell على pro-metaphase بيكون لسه condensation is taking place, but this is less condensed, which will give you more banding pattern. And if you have more banding pattern, sometimes if there is a small deletion, so for example, if you're looking على standard karyotype, وهاي الباند يعني بين صغيرة بس مش متأكد, you're not sure if there is a partial deletion in it. If you do high resolution banding, the same band will become into subbands, more bands in it. And this is high resolution banding. This is an example of chromosome 4. So A is a banding happened على 400 bands. B على 500 bands للchromosomes و C على 800 bands. So high resolution banding will enable you for the same band to see it in more resolution using high resolution banding when needed. يعني chromosome chromosome 4 Q arm region 2 subregion 1 ما بنسميها 4Q21 لا you call it 4Q21 region 21 can be divided into even smaller regions. Right, so let's talk about السنترومير والتلومير. All of the chromosomes عندهم centromeres. With centromeres is a satellite, repetitive DNA known as alpha satellite. What is alpha satellite? هو sequence of DNA طوله 171 bases repeated many times. So 171 bases نفسهم, copy and paste them next to each other many times, you will get the alpha satellite اللي هو centromere. Remember, centromere is very important, for example, during meiosis or mitosis, the proper cell division. If the centromere is missing or deleted, the entire chromosome will be lost during a cell cycle. And pericentromeric region اللي هي region surrounding the centromere. طبعا عندنا بعدين ال sequence of DNA that فيه genes وعندنا at the ends الtelomeres. Now التيلومير كمان repetitive DNA satellite if you want to call it and المونومر تبعه أو اليونت 6 nucleotides TTA triple G. All of the chromosomes, if you sequence the telomere with 24 different chromosomes, the telomere on both ends, they will have the same sequence, TTA, GGG, repeated many times. And if you zoom in, you will see what is written here, الجرين مكتوب السايز من 3 ل 20 كيلوبيز من 3000 ل 20000 nucleotide أو bases عبارة عن repetition of TTA GGG n number of times. After, if you go towards the centromere, عندي هون telomere associated repeated sequence, and then عندي in red unique subtelomeric sequence. Okay, so what you would do, so if you make a synthetic DNA sequence, هذا ال DNA sequence complementary the TTA triple G, and it is colored with fluorophorin green. And if you add it on the chromosomes, because all of the telomeres in all of the chromosomes, they have the same sequence, that probe or that synthetic DNA will show the green color on the telomeres on all the chromosomes. So a sequence of telomere is universal across the human chromosomes. The subtelomeric region is a little bit more specific to certain chromosomes. So if you see here a red color, لأنه sequence of subtelomere is unique لهذا الchromosome, with other chromosome and a different subtelomeric sequence. Now there is a point, as the cells divide in normal cells, when they divide, the telomere length will shorten. And when the telomeres shorten, this means that they will be lost and you will start losing the genetic material when you go into the inside the gene area. And when essential genes are missing, the cell will die. That is why in normal cells, our human, our living organisms, they have a limited life or division capacity because of telomere shortening, just like a tennis shoes, زي البوت بيحتروا, they erode with time. Exception is cancer. Once a cell is transformed into cancer, the cancer cells, they will reactivate an enzyme known as telomerase. التيلوميريز هذا, it will ensure that the telomeres are not eroded, are not shortened as the cells divide. That's why cancer cells, they can live forever, but they can divide indefinitely as long as they are provided with nutrients. Right, so if you take a cancer cell from a living or تاخد tumor biopsy من patient and you keep you add those cells, that tumor cells, in a petri dish or in a flask, and if you keep providing media with nutrients, they will literally live forever. But if you take a normal tissue from an individual, they will divide certain number of divisions, and after that the cells will die. Same with people. So when you age, and if you look at the chromosomes, you will see that the end of the chromosomes are eroded, aging. And there is a study on the 20s, with 30s, with 40s, with 50s, with population showing how the telomeres are eroding. So this is a double-edged mechanism. On one hand, the bad thing about it, it causes aging, or it leads eventually to finite lifespan. But the advantage of it, it will protect against cancer. ليش؟ لأنه when the cell starts dividing abnormally, more divisions than it should be dividing, something is wrong. It will start losing genes with telomeres and it will die. But if somehow the cell manages to transform into cancer, تحولت للزومبي للcancer, it will reactivate the telomerase and it will be immortal. This is another picture here showing you that even though telomeres, they have the same sequence in all of the chromosomes, the subtelomeric region is where you start seeing sequences that are more unique to a chromosome or certain chromosomes. And then if you go further inside towards the centromere, you will start seeing genes that are unique على different types of different numbers of chromosomes. And with this, we conclude today's talk. So let me stop recording. Do you have any questions? طيب تمام.